All right, where we ended, we were talking about the three different structures that are always going to be found in all of eukaryotic cells, at least at one point in time. The reason I keep saying at one point in time is we're moving on to number three, where we do find at some particular cells that this guy right here is actually starting to be lost. In particular, one cell, a red blood cell, as it matures, the nucleus actually disappears. It goes away. Now, what this nucleus is a double-layered organelle. Its main job is to house, kind of enclose the genetic material. It is also selectively permeable, just like we talked about with the cell membrane. It is choosy. It allows certain particles to go into and out of the actual nucleus. Now, the majority of stuff that's going in and out of it is probably mRNA or rRNA. The rRNA is the ribosomal RNA because inside the ribosome is this, or excuse me, inside the nucleus is this organelle, the nucleolus, that type of rRNA, and what its job is to do is to make this first R, which is ribosomal. What it's actually going to make, the nucleolus' job is to help make ribosomes. And as we learned in the last unit, those ribosomes' main function is to help in the production of proteins. Okay, chromatin, this is our next bullet point down, just taking a look at what this is, consists loosely of coiled fibers. We're going to get into the second half of this unit where we're looking at mitosis and the separation of the genetic material. That chromatin being, when you look at a cell itself, you've got your nucleus within it. So you can see it here. But a lot of times when you're looking at a cell, all you see is just this big blob. It's got a whole bunch of just dark structures within inside of it. Doesn't look very organized at all. What that is the chromatin. Now, once it's getting ready to divide, it needs to become organized. So the cell becomes organized, the nuclear membrane starts to disappear, and you start seeing your chromosomes become these little X's, which you remember seeing before. So it no longer becomes chromatin, it now moves its way into chromosomes. Okay? Alright, taking a quick illustration, take a look at this. We again zoom in real quick. You can see that you have your pores. Each of these pores are little holes that are going to allow for mRNA, rRNA to leave, and that those pores are going to be the phospholipid bilayer, which you've seen multiple times before. But I'm going to zoom in and look at the structure. You get all this little stringy stuff with inside there. All that stringy stuff with inside there is the chromatin, that genetic material. And if I didn't want that genetic material, I'll just kind of erase it here. There is a big section just right in the middle there. You can see it a little bit denser. That is the nucleolus. That's largely composed of rRNA. And again, that R stands for ribosome. So its job is to make ribosomes, and then so those guys leave, will either be found on the ER to make it rough ER, or will be transported throughout somewhere in the cytoplasm, where later on in some of the other biologies you might remember the whole process of translation, where the mRNA from the chromatin that has been condensed up will eventually find the rRNA and cause that conversion of moving the amino acids into place by a peptide bond, making your protein structure. All right, moving our way into the next section. So what we just covered now is all the stuff on cells and cell parts. Let's go back real quick. I'm just going to add a new slide. So if you think of the things that we had just remembered, that all the cells are going to have three components. Those primary components they're going to have is one, a cell membrane. Okay, they're going to have cytoplasm, and they're going to have the nucleus. Again, all of them are going to have nucleus except for the one that at maturity loses it, that being that red blood cell. Everything else is going to contain these three parts. Some of the specifics that we talked about with these three structures is that, again, the cell membrane is selectively permeable, so is the nucleus. What that means is that it's choosy. They can allow certain things to go into and out of it. The cytoplasm, its job is to basically hold organelles. Hold the organelles in place. Now, examples of organelles that we cover that you're going to want to know again are things like the ER. There are two types, rough, smooth. We've got lysosomes, we have ribosomes. Um, Again, nucleus would be one of those. Golgi body. Let's see. Vacuole, vesicle, so on and so forth. There's a number of them. And so what your job is to do is to kind of go through and identify what each of these guys are going to do. ER is transport. If it is rough, it has the ribosomes on it, which makes proteins. So rough transports and makes proteins. Smooth does not have ribosomes, so it's the synthesis of smooth and fats. And so it's going to help maintain and produce fats. Lysosomes to get rid of material, ribosomes to help make the production of proteins, Golgi body to package up those things that like were proteins or fats, vesicles to transport those proteins and fats throughout the entire cell, vacuole, nice storage site, so on and so forth. You can go into things like the cilia and flagella for movement. We can talk about the cytoskeleton and microtubules and microfilaments as well. All right, so that's the gist of where we're going within this first part, talking about the cell and the cell structure. All right, we're going to move into the next section. That next section is actually going to be dealing with movements with inside of the cell and the cell membrane. So we just talked about some of those structures before. Hopefully this stuff is pretty straightforward. So the stuff that we've seen is that, again, movement into and out of the cell. Now, the ways that this is going to happen is we've talked about in sophomore biology, simple diffusion. Diffusion is the movement of any particle going from an area of high concentration to a low concentration. And here I give you two different types of ways that diffusion or movements can occur within a cell. Number one is passive, and number two is active. Passive requires no energy. So there are, as shown there, three types without energy. Those three types are simple diffusion. Now, simple diffusion is going to be where molecules can pass through a cell membrane. We're looking at this thing before. Here's our cell membrane. We've got all of these little phospholipids in here. Okay. Remember, non-polar, polar phosphate groups. If I'm talking about simple diffusion, movement of any particle from high to low, those particles simply can pass right on through without help. That's what we're talking about in just simple diffusion. If I move my way into number two, which is this facilitated diffusion, that's this next one there. Facilitated diffusion is different. Say that I've got C6H12O6. Not very small. This guy needs more help. It needs a facilitator. It needs a helper. So facilitated diffusion is still going from an area of high to low, but it needs help, and this help is coming in the form of a protein. Okay? And the last one is by osmosis. Osmosis is still going from high to low, but what osmosis really refers to is the movement of water from an area of high concentration to an area of low concentration. So let's kind of move this around here a little bit. We're talking about movement of water or osmosis. If I take a beaker and I've got some liquid in it, let's make this kind of blue so we can actually have some sort of a membrane. Separate it out. Say in there, it's just H2O, pure water in here, okay? On this side, I'm going to pour in some sugar. Now, ultimately, we want to talk about diffusion. When we're talking about sugar moving, that sugar is probably going to transfer from where it is right now and wanting to go that way. I've got a high concentration of sugar. Put this over here. High concentration of sugar is going to want to go where there's a low concentration of sugar. So that's going to move over there. But where is there more pure water? pure H2O is over here. So I actually have high of H2O on this side, and it's going to go to where there's low H2O. So in fact, you could talk about osmosis and diffusion being different. It's where osmosis is the diffusion high to low, but of water, H2O. So osmosis is the diffusion of water, where simple diffusion is just high to low. Okay, so high to low of this case, in this case, a sugar that's being diffused. Does not need a helper. Now, if all these were blocked and these sugars are too big and I needed some sort of a protein or some sort of a door to get through, that would be considered facilitated diffusion. Okay. Simple examples, we talk about it, look at it if I'm rollerblading, I'm on top of this hill, right? Doesn't take a whole lot of effort to go down. Doesn't require a whole lot of energy. But if I'm at the bottom of this hill and I want to rollerblade or bike or run up, you know that it takes a heck of a lot more effort. And that's what's going to move into our next set, which we're talking about here. Way over here, you see it? This next set is active transport. Active transport actually uses cell energy in the form of ATP, which the mitochondria make for us. All right, so its job is to make active energy. So in our sense, if we're looking at how this is actually going to work, let's kind of move this stuff off to the side. We are going from an area of low concentration. So if I'm going to draw this out, if I have sort of a beaker and I've got all these sugar molecules or salt over here, and I've only got one over there, I want to force one more over there. It's going to require energy. That's active transport. In order of saying it from low to high, you're on the bottom of the hill, you want to get to the top, you require a lot of energy to get there. So you're actively working, active transport. All right. So putting that in terms, we just mentioned diffusion is the random movement of particles from an area of high to low. Facilitated is the same thing, but they are too big, so they need a helper. So they move across the cell membrane with the use of a protein. So again, those big helper cells that are going to be found in the middle, okay, that allow the molecules to travel on through. Here's just a quick illustration of that. You can see by this structure here, let's use a fun color. So it's actually moving on through. Let's use green here. And it's actually going through that protein, so that's facilitated diffusion. Now, if this little guy right here can move its right on its way through those little Dippin' Dots that phospholipids there, that would be simple diffusion. Here, if I'm going to talk about just pure water moving back and forth, that's where we're going to get into osmosis. Remember, osmosis is the diffusion of water, high to low of water. So, I don't want to confuse you. We can come back to this later on, but the easiest way for you to remember is that water, where there is a lot of water, there is little solute. Okay, if we go back to our illustration, where I have a lot of water, so if this is my sugar, I have more H2O over here, which means I have less sugar on that side. So where there's low solute, solute like sugar, to a high solute, so the water is going to want to rush this way. That's osmosis, where it's high to low of pure water or low to high of a solute. Okay? I think what we're going to end up doing is stopping there for today. I'd like to talk to you guys about the hyper, hypo, and isotonic solutions in class. So you guys can then take the rest of the hour and start working on your cell structure and function packet. See you tomorrow.